A very good morning to one and all, respected dignitaries, faculties, and dear friends. Today, the Department of Physics at St. Thomas College is conducting a national webinar on the topic Natural and Synthetic Nanostructures Applications in Healthcare. Before we begin, let us invoke the blessings of the Almighty with a silent prayer. Thank you. St. Thomas College Autonomous, Thrissur, has been one of the leading academic institutions in higher education sector of Kerala for over a century, with the Department of Physics itself being one of the oldest departments in the college. The department has been actively conducting a series of webinars and lectures on wide variety of topics aimed to develop research interest among its students. Today, the department, in association with the IQAC of the college, is proudly organizing a webinar on one of the most intriguing areas of research, nanomedicine. With that said, I would like to invite Professor P.M. Joy, Head of the Department of Physics, St. Thomas College, for the welcome speech.
Respected Principal Dr. Joy KL, the resource person of the day, Dr. Asif Khan Shanawaz, convener of the webinar, Dr. John Snadwas, my dear faculties, and my most beloved students. Today is a very precious day as far as the physics department is concerned, because we are conducting a webinar on natural synthetic nanostructures and application in healthcare. That is in the field of the nanostructures. Actually, the nano field is related to the core subject physics. Our principal, Dr. Joy KL, is always been an inspiring and catalytic person for all the activities of our department. He always encourages us with the maximum effort. On behalf of the Department of Physics and on behalf of all the faculties and students, I extend a warm welcome to Dr. Joy KL, principal of our college. The resource person of the day is Dr. Asif Khan Shanawaz, who is working in Institute of Nanoscience and Technology, Mohali. He took his MTech from Amrita Institute of Technology and PhD from IIT Bombay. He has 20 publications and 2 patents for his credit. I think he is the very apt person for conducting this seminar. On behalf of the management of St. Thomas College and on behalf of the Department of Physics and on behalf of all the faculties and the students of the department, I extend welcome to Dr. Asif Ali Khan Shanawaz. Okay, I take this opportunity to welcome all the faculty members of the department and all the students from inside the colleges and outside the college. And also, I welcome all the faculties of the other department. This is a very rare opportunity as far as the students are concerned, because a scientist of the international standard is interacting with our students, and you can have a lot of varied experience from him. I appeal to you to make maximum advantage of this situation. Once again, I welcome all the faculties and students to the program. Thank you. Thank you very much.
Thank you, sir. Now, let me invite Dr. K.L. Joy, Principal, St. Thomas College, Thrissur, for the inaugural address.
Okay, very warm good morning to all. Resource person Dr. Asif Khan Shanawaz, INST, Mohali, Professor PM Choi, the head of the Department of Physics, Dr. Joe Karakudan, Assistant Control Examinations, Dr. Jones Naduth, Ms. Anubama, faculty members, research scholars, and my dear students. It is highly appreciable that our research department of physics has come forward to conduct an international national webinar on natural and synthetic nanostructures application in health care in association with the IQAC. In recent years, a new field known as nanotechnology has impacted multiple industries by taking advantage of the special qualities of these small atomic sized particles. Nanomedicine has already opened up a new avenue of research in cancer therapy, drug delivery, and immune regulations. While the benefit of this new science to human civilization are seemingly immeasurable, it is also important to appreciate that these particles can also lead to harmful effects on human health. We are sure that today's our resource person, Dr. Asif Khan Shanawaz, a scientist, chemical biology unit, INST, Mohali, will enlighten us with the flame of knowledge he has experience in nanotechnology. All-hearted congratulations to Professor PM Choi, the head of the Department of Physics, Dr. John Snudworth, and team for organizing this national webinar. I hope you will find this webinar highly fruitful and beneficial for your future work. Wishing everyone all the best. With your permission, let me declare this national webinar open. Thank you. Thank you very much.
Thank you, sir. Now it's time for the much awaited technical session. I would like to take this opportunity to introduce the resource person, Dr. Asif Khan Sanavas, to all our participants. Dr. Asif Khan Sanavas is currently a scientist at Chemical Biology Unit, Institute of Nanoscience and Technology, INST Mohali. He did his graduation and post-graduation in biochemistry from PSG College of Arts and Science, Coimbatore, followed by MTech in Nanomedical Sciences from Amartya Center for Nanosciences, Cochin. He did his PhD in biomedical engineering from IIT Bombay. His research interests include targeted combinatorial nanomedicines, organic inorganic biomaterials for theranostics, and plasmonic nanomaterials for photothermal therapy. He has many publications and patents to his credit and is a recipient of many awards, honors, and fellowships, including the DBT Indo-Australian Career Boosting Gold Fellowship. We feel privileged to have such an esteemed guest speaker for our humble program. Before we begin, a word to the participants. Please feel free to post your queries in the live chat box. We will be having a Q&A session towards the end of the lecture, and the link to the for the feedback form will also be posted in the live chat box. I hope everyone would make the most out of this opportunity, and thank you. Over to you, sir.
Thanks, Mrs. Anupama. I hope I'm audible. So, okay, so I'm just, I hope everyone can see the slide showing natural and artificial nanostructures. So, you may have come across various talks on nanotechnology, and I'm sure that This is not something new among the audience today, because I heard that one of the stalwart in the field, Professor T. Pradeep, gave a lecture some time back, at St. Thomas College through online mode. I'm sure he would have given a fascinating lecture. Today, just to have a bit of difference from a regular way of presentation. To start with, I'm going to have a video presentation where I have put together some online videos. The credit is not mine. I have taken it from online resources. To first show something which is kind of inspiring us, the nanotechnologists, from the nature, from various structures that are present in nature. And I don't want to give too much of the science that I do. I will come to that only towards the end of this lecture. Rather, I will talk more generic about the topic and how we can get inspired by the nature and its the various structures that are present in nature, and how we can use them for our benefit, especially the biomedical or clinical or healthcare applications. All right. So to start with the first presentation, you are seeing in this particular, in the screen, you're seeing a nano robot which is trying to treat a cell which is malfunctioned, which is not functioning properly, or probably it is kind of trying to kill a cancer cell that is present in the blood, which is present in the bloodstream. You are seeing a lot of RBC cells, around this particular cell. This is a vision for the past many decades. It's not just something that I'm showing today. The scientists around the world have been having a vision of such an application of nano or micro robots, which can actually crawl inside our body, go specifically to different parts of our body, and it can cure something that a regular conventional therapeutic procedure cannot cure. But as I said, it's like more than 50, 60 years since we invented nanotechnology. Mid of 1900s, we started coining the name nanotechnology. But prior to that, in 1800s, Faraday had already invented it, but not exactly knowing that they are nanoparticles. He already invented it. So for the past decades, we have been inventing new things in nanotechnology, especially in case of clinical applications. Such nano micro robots are something still a dream that yet to come true. But as we know that nothing changes overnight, right? I mean, we need to step inch by inch towards a particular dream, and this particular presentation is to show that. So before talking about the nano robots, we need to talk about a recent Nobel Prize award that was in 2016, that was given to these three scientists: John Pierre Savage, Sir Fraser Stoddart, and Bernard Feringa. So they three got a Nobel Prize in chemistry for their invention on molecular machines. All right. So, but then they didn't do things together. You will always see Nobel Prize given to more than one person for a particular invention, but they wouldn't have worked together. They always work separately in their own labs. They may be having different way of approach to a particular problem, but everything converges to the same problem, right? So they all were working on something that actually can realize the dream of nano motors or nano machines or molecular machines. So they actually were kind of focusing on creating molecular machines which can actually voyage through various fluids, especially the blood in the future, as you see in the screen right now. So these molecular machines are something that are of the size thousands of width of the human hair. That is, they are very very small. And to give a sense of what exactly it is, you can see a skin, and when you go down over the skin, you will start seeing dead cells. You will start seeing crevices, and you will start seeing cells. And when you get into the cells, you will be able to look at finally the DNA that is present in the nucleus. And the width of the DNA is about a nanometer, and you can see the number of atoms that are present within 1 nanometer. So that is the sense of nanometer scale that one has to think of when we are talking about nanotechnology. So to come to the molecular machines, the first person that among the three, John Pierre Sauvage, he made something called ring-shaped molecules, which resembles chains in a motor. So now, instead of talking about a molecular machine or a motor, let us talk about a motor that runs something, right? So let us just imagine a car that has chains, or that actually connects the motor to the wheel, and in the basic fundamentals that we have it in the cycle that we pedal. We actually have pedals that actually runs the wheel, and we are the pedal and the wheel is connected to the chain, and there is a mechanical motion that is involved. And in case of motorbikes and cars, we have, instead of pedal, we have a motor, right, which actually mechanizes the wheel through its own dedicated mechanisms. So let's just assume that these three scientists invented three different parts of a car, which, when put together, can actually provide functions of a nanomotor. So John Pierre Sauvage invented molecular chains which independently can link together, similar to the chains. And the second person, Fraser Stoddart. So Fraser Stoddart made a ring shuttle that actually it resembles a molecular axle, and you can see a macrocyclic molecule, and there is a dumbbell-shaped molecule which goes along the axis of this macrocyclic molecule. So it resembles something called something in the car, which is the axle of a wheel, which you can see there are two wheels along the axle. And this particular mechanism, this particular invention, Sir Stoddart used it to have a nanomolecular lift. You can see there are three axles, and each one of them is powered by electricity. And when there is a charge given, there is a moment of this axle, this molecular lift moving up. So this is something that he invented, that which actually fills in the puzzle for another component of a car. We need an axle and to run the car. Now, coming to the actual motor, right? So when we are looking at motors, in a car, for a nano machine, we are looking at the work of Professor Ben Feringa, who actually contributed to the heart of nano motor. So he actually invented, structure of molecular, structures which can have a four-wheel drive. And as you can see, this is invention that actually, when put together with the other two works received the Nobel Prize. Now, when we are talking about nano machines, artificial nano machines, which attracted the Nobel Prize, it was nothing that new to the nature. We already have various molecular machines that are present in our body, in various other organisms like bacteria. So we have much more complicated molecular machines inside our body that is allowing us to function, to live and function. So so whatever the work these three scientists have done is very, very, very small compared to what we have evolved into in case of these molecular machines, right? So over millions of years, our body has evolved into this complex machine that doesn't function isolated. We are not isolated beings. We are beings of multicellularity, and each of the cellular components are living by itself. They are moving, and they are energizing us to move ourselves as a big multicellular being. So to give just a couple of examples of these molecular machines present in our body, we are taking two machines that are present in our body. So I just introduced you into something artificial, that is nano machines. We will come to that later at the end of this video presentation. We will come to that later. How these nano machines that they have made are tested, and what are the other nano motors that are also invented artificially. But to have a sense of sense of understanding or sense of attachment to the work that these people do, we need to understand ourselves, our own mechanisms that is going on inside our body. So I hear that the audience today are undergraduate students, postgraduate students, and PhD students. So I think this is relevant to everyone. We need to get inspired by the nature itself. So coming to the first molecular machines, that is ATP synthase, which is the macro molecular motor that actually energizing each cell by synthesizing something called ATP, which is adenosine triphosphate. So this ATP synthase is basically present on an organelle called mitochondria. So all of you, irrespective of your current field, you would have learned that in your +2 in your school. So mitochondria is the powerhouse of a cell. So how does it power the cell? Is through these ATP synthase. You can see so many of them in the mitochondrial membrane. So to know how ATP synthase works, you can actually relate that mechanism of ATP synthase to this DC motor. You all know what is a DC motor, direct electricity motor, direct current motor. So in this particular motor, we have two components. One is a rotor and a rotor and a stator. So you can see that this rotor, according to its name, it rotates along the axis of the stator. So the stator basically have a lot of solenoids which has magnetic field. Each of them has its own polarity, and it drives this rotor to rotate on its axis. So here we are looking at converting electricity to mechanical energy, right? Electrical energy into mechanical energy. So similar to a stator and a rotor, in case of ATP synthase, you can see a rotor that is rotating along a stator which is on the bottom. So this stator is something that keeps on moving, and it ends generating ATP molecules. So now, how does what do you mean by generating an ATP molecule? Our body has a lot of mechanism that molecular metabolic reactions that that requires energy. So we are driven by energy. The whole universe is driven by energy. So at molecular level inside our body, the energy actually comes from something called ATP, adenosine triphosphate, and the energy in this particular molecule is stored in the phosphate bonds. There are three phosphates, and the end phosphate molecule is the molecule that when it breaks, the broken bond actually is converted into the energy that is required for various reactions. So when the ATP is converted into adenosine diphosphate, when one phosphate is removed for consumption of energy, this ADP again gets recharged to ATP through the ATP synthase. All right. So basically, we have a partially charged battery, which the ATP synthase converts into a fully charged battery. Now, so this particular, each of the bond that is present in the phosphate has a stored energy, and when it is broken, as I said, it releases the energy for a particular molecular mechanism to be driven and to be completed. Now, coming to the actual ATP synthase, I have to just wait. Oh, yeah. So here you can see the rotor, which is rotating. There are multiple units on the rotor that that actually makes the assembly of the rotor, which is embedded in the mitochondrial membrane. And when there is a proton influx between the membrane of the mitochondria, so there is the generation of this ATP through the electron transfer. So you can see in the stator there is ADP coming in, and it is binding to one of the subunit of the stator, and it gets energized. So when the rotor comes back, it opens up this subunit and releases the ATP. So this ATP, so many of the millions of them are being generated by the ATP molecules, ATP synthase, present on the mitochondria. So there are so many mitochondria within the cell, and each of the mitochondria has so many molecules of these ATP synthase, and it eventually generates a lot of ATP. Now coming to the second molecular machine, which is kinesin. So kinesin actually, again, it is a molecular machine, but it doesn't generate energy. Rather, it consumes the ATP that is generated by ATP synthase, and it functions by carrying different cargo molecules across the cell. So here you can see. So here you can see there is a central component, which is nucleus, and there is a lot of strands that comes out of the nucleus, which is called the cytoskeleton. And this cytoskeleton not only helps in keeping up the shape of a cell, but also helps in movement of various components across different organelles present within the cell. So you need to communicate. Each organelle has to communicate between each other so that there is coordination between them. There is any exchange of chemicals, signals between them so that the cell functions properly. So here you can see a kinesin molecule carrying a big vacuole to be transferred from one end of the cell to another end of the cell. So when we look closer at a kinesin molecule, it is something similar to how we walk. It has two motor heads that actually walks like our 2 feet, how we walk one step at a time. And it walks on this microtubule, which is part of the cytoskeletal system. So when we look at the kinesin molecule, we are looking at two motor heads, which are, as you can see, which are kind of moving, and these motor heads has two catalytic core. So these are the core that binds to ATP. These motor heads are connected to the coil to the by through the linkers, and they walk on this microtubule. It is something like a road or a track for them to walk on, and this coil to the connected to the motor head is connecting this motor head to the organelle, which can be anything, which can be a vacuole or any cargo that it wants to bind. The catalytic core initially binds ADP, sorry, it binds ATP. Now it has released the phosphate, so it now has ADP in it. So when it has ADP, it releases the ADP, and it binds a new ATP molecule. So when this ATP molecule is bound, the free motor head is undergoing a conformational change, that it is thrown forward to bind to the microtubule. So once it is bound to the microtubule, the previously bound ATP is converted to ADP and free phosphate. And this new motor head that is bound to the microtubule now binds a new ATP, and this process keeps on going. So ATP binding is important for the conformational change, so that the motor head can step forward and move on the microtubule. So this is a very complicated evolutionary machine that the nature has developed, and this is something that is a very fundamental process which is important for the cell to function. Unless this functions, there won't be signal transduction happening inside the cell. Chemical transduction, signal transduction, everything, all the molecular metabolic reactions that is happening. So it is very fundamental, important, vital, reaction or mechanism that makes this function. So ATP synthase produces ATP, and these molecular machines like kinesin uses that ATP to function at a very fundamental level, and keeps the cell running, living, and functioning, right? So these are something that are present in the nature that helps, for various machines, whether all living machines to function. So coming back to the artificial molecular machines, which we initially looked at, we left where we had a nano motor, which, as you see here, we have in the gray color, the wheels of this nano machine, and it is connected by an axle, and there is a chain that connects these axles, and it is kind of the most fundamental artificial molecular machines that has been invented. So how do you test a molecular machine? We are talking about very small structures which cannot be tested normally. We need extreme conditions, and we need inert surfaces. So what inert surface that they use to test them is gold substrate, as you see here. These nano machines, they can be tested on these gold inert surface, because there won't be much of resistance or any kind of bonding that will be created between gold, it being inert, and that way these nano machines, which are called as nano cars, to move freely on the track. Now, again, just because you have a track and a nano machine, it doesn't actually move along. You need extreme conditions, as I said. So this particular gold nano substrate is kept inside a very highly sophisticated instrument called scanning tunneling microscope, which actually maintains a very high vacuum and inert condition, so that there won't be any reaction from the atmospheric, the surrounding atmosphere of the nano car to have any inhibition in its movement like this. This is a simulated movement of a nano car on a gold substrate. You can see individual a gold atom, and and you can look at the scale of these nano cars on the array of gold atoms that are constituting the substrate. So we have to this scanning tunneling microscope, when can actually do two things. It can actually propel, or it can actually stimulate these nano cars to move on the substrate, and also it provides very good inert conditions for the nano cars to move. You can see it, the screen of the computational rendering of these microscope provides the idea about whether the car moves on these substrates through this particular images. And you can make different types of these nano cars. You can see they have different structures, but they all end up moving when the probe on a scanning tunneling microscope is kept very close on them. You should not be touching them, rather you keep very close proximity to these nano cars. So there will be a Newtonian forces, very small forces that can actually propel them, that can actually push them to move further on the track of nano cars. And these in fact, based on these nano cars, there was a nano race that was held also among various groups across the globe. So as similar to a car race, F1 car race, we all know about. Those car races are just to advertise various companies, right? You can see so many companies, companies that manufacture wheels, tires, cars. So they all actually invest on these F1 races because they want to advertise their companies, market their companies to a wider audience. It is, a car is something that is synonymous to speed. So the race actually provides that sense of speed and car, the synonymity, and advertises various companies. So similar to the such advertisement oriented F1 car races, here they wanted to advertise their groups. They wanted to show their expertise in making molecular machines, and also to make the researchers across the globe excited that it's not just about research. There's so much interesting that goes around these research, that you can actually have competition among different groups, a healthy competition to showcase what they can make in their labs. So I think the Swiss research group won the race, and they were able to move about 100 nanometer, if I'm not wrong. So you can see in case of these nano cars, their movement will be limited to a very small. Right now, it is limited to a very small size or a length distance scale. So coming to other types of nano machines, the researchers across the globe have actually made different nano machines, and they can actually propel through liquids. As you can see in this particular video, it can actually propel through a liquid where it can generate different types of. 1 minute, I think I have missed something here. So this particular nano motor is able to generate add bubbles. That these are basically the reaction of these titanium oxide or photocatalytic nano motors, which can actually, in the presence of hydrogen peroxide, produces hydrogen gas, which can propel these nano motors further, so that they can, in the front you can see a cargo which they can move around. So this another motor is it's a magnetically controlled motor, which is also comprising of MOF. It is writing the letter MOF, MOF, which is metal organic framework, and it is constituted of metal organic frameworks. We will come a bit on this metal organic frameworks, very small glimpse at a later stage of this presentation. And apart from these molecular machines, there are various other molecular machines that are also prepared by various researchers. Most of them are Janus particles. Janus nanoparticles are particles which, the name of them is derived from a Greek letter Janus, which means two-faced. As you can see in the center, there are two phases. And apart from looking, um, uh, uh, looking different from two different sides, they also have different functionalities on these two sides. And because of these functionalities, they can actually contribute to nanomotor research. So these motors can be driven by various stimuli: light, thermal, that is temperature, acoustics, sound, basically, and magnetic stimuli, chemical stimuli, pH, electrical, so on and so forth. So all these stimuli propel them, make them run under a particular defined conditions. So coming with this, I'm kind of finishing this particular video presentation. To give the credits to all the videos, you can see where these videos have been taken from different uh, different quarters of the online resources. Now I'll stop this video presentation, and I'll move on um, to to the slide presentation. I'll just share that first. So just taking some time to. Fortunately, it's kind of. So there is some issue. I can't see the share option after clicking the share screen. Uh, Dr. Jones, can you share it from your end? Is it possible?
Okay, I will share from my side. Okay, 1 minute. Just wait. So just tell me when you need to change the slide. Hello?
Yes, I think
Hello?
I can share it now.
Okay, okay, okay. You just share.
If that is okay, I will try to control from here. All right. So
Okay, okay.
now coming to the second part of the presentation. So Miss Anupama, can you just confirm if you are seeing the presentation?
Yes, sir. Please, could you, if you could make it into full screen mode? Sir?
Yeah. Can you hear? Can you see it?
Yes, sir. The presentation is visible,
Okay,
but
all
if
right.
you could
So
make it
I
into
can't
full
hear
screen
you.
mode, please.
I guess you can see it. All right. So 1 minute. I'll just. So the next part of the presentation is nature-inspired nanotechnology to solve biomedical problems. So in the first part, we looked at how we can make molecular machines, which are something that we already have in our body, but we can also make them synthetically, and that is basically to show how advanced we are getting in case of nanotechnology, and how we are able to
Asif,
uh, you know
Asif,
uh, we are able
Asif,
to
can you
get
hear me?
inspired from the nature, and how we can solve decades long
Asif,
problem of having a nanomotor running
Asif,
through our body. But there are other natural nano technologies that which are being adapted by various researchers for solving various biomedical problems. Specifically, there are various other problems they are trying to solve, but I will be talking only about the biomedical problems, how we can have solutions. So prior to looking at those, let us just have a very glimpse of what is nanotechnology. I'm sure you have some basic idea about nanoparticles, the nanoscale of it. My friend Dr. Jones, we shared the same period of study at IIT Bombay, and he would have also taught some of you about nanotechnology. Just to mention, he's one of the topper. He was one of the topper during his stay at IIT Bombay. Okay, so now coming to what exactly is nanotechnology. So we are looking at it is basically an enabling technology. So it constitutes of creation and utilization of materials, devices, and systems on the nanometer length scale. Right? Nanometer length scale constitutes of atoms, molecules, and supramolecular structures. What is supramolecular structure? Hello, hello, hello. Full screen. Okay, ipo full screen ayo. And it is full screen on my side. You can see now? Okay. Okay. ഇപ്പൊ കാണാൻ പറ്റുന്നോ? Now is okay. Okay. But you can hear me, right? Okay. All right. So sorry, there is some issue with the full screen. From my side, it is full screen, but I don't know why is it not showing up there. But let us get back. So, as I said, it is an enabling technology, and it encompasses all major disciplines of science and technology. So, it involves chemistry, which is important to synthesize these nano materials. It involves physics, where you need to understand the physical characterization you have to do so that you can understand various properties of physical properties, chemical and physical properties of these nanomaterials, and you can actually have biological applications of these nanoparticles, and engineering applications of you can engineer them, and you can have various technological applications of these nanomaterials. So as I said, we can make molecular tiny motors, and there are carbon nanotubes which can enable different types of applications in engineering and technology. Now, as I initially said, it is a very old technology. The only thing is, it was not realized till 50, mid of 1900s, and a few decades later by a set of scientists. But then in the mid of 1800s, more than a century before, Michael Faraday actually made this gold colloids. He just didn't know that they are nanoparticles. But even before him, in the fourth century, the Romans had made this gold colloids. They actually used it to dye their hair. They used it to paint various glass and other structures to give different color combinations. And you can still see these glasses in various churches in Rome, in France, in Italy, Rome. Basically, it's a very age-old technology, but only recently we realized them as nanotechnology, right? And the father of the person who is who we know as a father of of the nanotechnology, Richard Feynman. He had established the basic concept, and he was able to coin the term, the phrase, "the plenty of room at the bottom," which is very famous in one of his lecture. And he is basically an inspiration in the field of nanotechnology to have various futuristic applications that we can have with this particular technology. So nanoscale, when we have nano particles made from bulk materials, there is a significant property difference that we look at when we miniaturize these particles. We have more functionality. They get more high surface area to volume ratio, which uh, which actually constitutes the basics of these nanoparticles for various various applications in drug delivery and in gas storage, etc. So they actually have novel phenomena when they are cut down to very small nanoparticles and particles, and their physical properties, their melting point, their reactivity, their conductivity, their mechanical strength, and their optical properties. All these actually end up becoming different when. Okay, so all right. All these become different when we are cutting down bulk materials into nanoparticles. So moving on. So as I said in the last presentation, I was talking about metal organic framework. So that is one of very exciting nanomaterials that are coming up, which can improve high, gas storage and drug delivery applications, because it is highly porous and it can constitute of very high surface area to volume ratio. So the significance of these types of materials is that they can, within a few grams of these materials, have high surface area, that their surface area is equivalent to a whole football field, right? So much of high surface area that can be generated by these nanoparticles, and because of this, you can have very high volumes of gas storage, very high loading of drug molecules within nanoparticles. So there are so many applications that you can come up with for nanoparticles. Apart from this surface area increase in the surface area, we can make them with defined shapes. There is infinite possibilities in making them in different shapes. As you can see here, you can make them in spheres, rods, bowls, or cups. Going beyond making a bowl, these researchers who made them have put a small sphere of nanoparticles inside each of these cups. You need how much tailoring, how much definition that you can bring in to making these nano shapes. You can make them cubes, stars, orthorhombic pyramidal structures, octahedral structures. So many applications, so many possibilities of making these structures. And when you are making different shapes and structures, their optical properties differ. So in case of gold, they are exclusively used on photonics, nanophotonics, and as you can see, spheres, pyramids, rods, and shells. These are not spheres; rather, these are shells. There is either a hollow core or there is a dielectric core. And on top of it there is a gold shell. So each of them have defined absorption in the electromagnetic spectrum. So the visible region, it can absorb in the visible region here, and from 650, 700 onwards, we have near infrared region. There are different windows, near infrared 1 and 2 windows, which have defined applications in biological, biomedical field, where bodily fluids absorb a lot in visible region, so you cannot use light in this region, but you can use in NIR region, where water, hemoglobin, and other components in the blood, in the tissue, doesn't absorb. So you can have easy or better penetration of light into the body. So we'll talk about this phenomenon, where we can use photo-assisted hypothermia or photothermal therapy in the later part of this particular presentation. So coming back to nature, nanostructures in nature. So how different natural structures are basically nanostructures. Okay, so we are talking a lot about iron oxide or magnetic nanoparticles. We are using them for right now. We can synthetically make them with different routes, chemical and dry and wet chemical methods. So, but in nature, these nanoparticles, magnetite particles, are made by specific bacterium, and they use these magnetic nanoparticles for their migration. So they actually look at the orientation of these magnetic nanoparticles to the Earth's magnetic field, and they migrate in their micro environment using that particular field. So migration based on intracellular magnetic structures, they are called magnetosomes. So they are present within a membrane. They are made within the membrane. So you can see nature actually can create magnetic nanoparticles. It is not new that we can only synthesize them. So in fact, these bacterium making these iron oxide or magnetite nanoparticles, they are also used in some biomedical applications, and also to understand how these bacteria move under different strengths of magnetic fields. So it gives a lot of insights about biological applications of magnetic nanoparticles. So moving on to a very basic observation that we see from
Sorry
our
to interrupt you, sir.
childhood days. We see these
Sir?
water striders. They walk on water, right? They don't sink. Apart from Jesus Christ, nobody else can walk on water, right? We can't. So in nature, we can see a lot of insects actually walking, but there is no divinity here. There is more of science behind this. Insects walking on water. So there is a basic science behind this. These the legs of these water striders, they have micro hairs, and each of the micro hair have nano grooves on it. So they these micro hairs will be able to spread the weight of these. Hello. Desktop. Okay. Upon another. Okay. 1 minute. I'll just share the screen. 1 minute. I'll share this. So you can see the screen now. All right. So now I hope things will be better. So these water striders, they have these micro hairs, and they have nano grooves. As I said, when we are looking at nanostructures, they have very large surface area, and these nano grooves can have a very good amount of air molecules trapped inside these nanogrooves. So this will give buoyancy to the insect, and the water surface tension and is also used to have stability for these water striders on the water on the liquid surface. So they also have very small surface forces, van der Waals forces between the water molecules and these micro hairs, so that they stay stable. They don't actually sink as well. So moving on to next type of structures, these are spider legs. When we look very close at them, they have these small hairs, which actually helps in the suction of the spider's legs onto different surfaces. You can see them walking on the ceiling without falling down, right? So how that happens? There is each of this micro hairs have its own van der Waals force in bonding chemistry, right? In bonding chemistry, van der Waals force are the weakest forces, right? They are the weakest forces. But here, with these spider's legs having van der Waals forces, they can actually have the strongest binding to various surfaces. So how does a weakest force provides the strongest adhesion to various surface? Here, each of these hair, there are millions of them. Each one of them actually have its own van der Waals force. When millions of them come together, okay, so I think I will have to zoom this normally and again. So millions of them, when it comes together, it gives a lot more a stronger adhesion to different surfaces. So why we need to get inspired with such very strong adhesion? Can we have in the future super strong adhesives in surgery and trauma? So what do you mean by trauma in clinical setting? Trauma is basically, if there is excessive bleeding through open wounds, through an accident, a road accident or any other accident, or even in military setting, you have a bleeding that happens and it needs to be stopped immediately before the person is moved or lifted to a nearby hospital setting. So can we have something that immediately seals the wound by sealing or sealing the skin that actually split open, right? And in surgery, the surgeons, they cut various parts of the body to look into and do some surgery, and then during that time they might cut different blood vessels. Can we glue them together by adapting to these kind of structures? Can we have some tapes that actually put onto these structures which are super hydrophobic, so that they don't actually get wet due to the blood, but becomes more adhesive in such very non-favorable conditions? So moving on to other adhesive structures that are present, natural structures. You see, apart from spiders, there are smaller smaller or insects which also cling on to different walls and ceilings, and then bigger organisms like lizard. So as you see, when the complexity or the weight of the organism increases, the structure becomes more and more complicated, more and more nano structured. So in case of beetles, you can see uh, they have microstructures, but when we become more and more complicated, in case of gecko is a lizard which has even more finer nanostructures that help in their in binding on to different different surfaces, and of course the same van der Waals forces help in keeping them stable on different structures. So adapting these gecko adhesives, various researchers have come up with different adhesive, artificial adhesive surfaces which can help in, as I said, gecko tapes, which can help in, sealing different wounds, different traumatic wounds and surgical wounds. Now, moving on to how natural nanostructures can provide strength to different, for different applications. So here we are having a bird which is a token. Token have such a massive beak. This beak is used to make holes, something similar to a woodpecker. So they help in crunching onto harder food. They help in the beaking of the wood for making nests. So you for such high energy, high tensile mechanical applications or utilization, it requires a very strong beak, and this beak basically is made up of keratin which is glued together. These are keratin scales which are of 50 nanometer, 50 micrometers, and 1 micrometer thick. And when we are looking even closer, they are having nano porous structures and they have a bony fiber and drum-like membranes, and they have a very high Young's modulus, which actually is required for applications in um, uh, in aero mobile industries, right? So where we need very high tensile structures that makes these aeroplanes and jet flights. So moving on to nature-inspired biophotonics, we see a lot of these butterflies, hornets, and beetles and moths, very colorful, insects that we see around. But fundamentally, they are not made up of any chemical dyes. These colors are not coming from any small dye molecules, but they are actually coming from their nano structured wings. All right. So they basically have colors and color variations because of nano patterns that are present in them. These patterns are provided by the ridges that are the pores and ridges that are present on these wings. So the ridge basically have a refractive index, which is different from the refractive index of air that is entrapped inside each of these nano ridges. So when there is a refractive index change in refractive index, you end up having a quarter wave system. I am not an expert in these topics. You can look them up. You basically the physicists that are following, that are in the audience, you will be able to understand these systems better. So these the light incident at narrow angles produces multiple reflections from these stacks. So this is only one stack, but they actually end up stacking together, and when they have multiple reflections, resulting in multiple constructive interferences, and these the interferences increases in the net reflectance, which provides different light, which hits some when it hits our eye, it provides different light. We actually see different light, different colors. So how it can be used in biological applications? Very recently, a paper that in they in Nature Nanotechnology paper, they actually got inspired from butterfly wing nanostructures, and they were able to make sensors which were tested for sensing intraocular pressure. Ocular is eye. So there is a lot of liquid pressure that is generated inside eye. So this pressure sometimes in some cases, because of too much of pressure or less pressure, there will be there will be a pathological conditions a patient may not be able to see. So they made a sensor that can continuously sense the intraocular pressure. They tested this in the eye cavity of rabbits. So you can look this paper up and you will get to know more. So I was talking about super hydrophobic surfaces few slides back. When we were talking about Gecko tapes. So these super hydrophobic surfaces helps in keeping water molecules away from the tapes when we were talking about Gecko tapes, right? So they should not get wet because when they get wet, they will lose their adhesiveness. But apart from that, these super hydrophobicity is also inspired from um, shark skin. You might have seen that shark skin is very, very, very clean compared to a whale's skin. You can see a blue whale having a lot of mollusks and other fouling sea creatures clinging onto them, and they that makes them completely not so beauty, and in the bottom especially, because these are opportunistic mollusks which cling onto these blue whales, and also they get to move from one place to another, and these are opportunists in the nature. In case of shark, you will not see these bigger organisms or even very small bacteria growing on their skin, because they have a very, very specialized skin scales, which gives them a grooved scale, which first of all gives a very good movement of water, and this movement of water doesn't allow them to have any fouling agents, marine fouling agents to bind onto them. So bacteria, algae, or any other mollusks. So based on this shark skin, a company actually was created, and in fact they tried to mimic the pattern of shark, and they were able to reduce growth of bacteria on these different nanostructures, nano patterns that they could form inspired from the shark. So you can see only at a particular pattern, they were able to avoid bacterial growth. But when they were changing the pattern, you can see the pattern, the bacterial growth could actually was phenomenal. They could colonize. So a very specific nano adaptation of these shark skin can provide different applications. So if you want bacteria to grow, you want a particular design pattern. But if you don't want for particular applications, you can have a different pattern. So apart from that, we will move on to how nano and microfluidics can help in biological applications. So we will talk about chemotherapy, nano drug delivery a bit later, but we can have different microfluidic chambers deliver drugs at a very precise times, and they can actually have different micro channels which can disperse drugs, and through micro needles they can be injected. So these micro needles are something that is dissolvable, and it is also inspired by the needles, the proboscideum of the mosquitoes, which injects into our bloodstream. It sucks up blood, and inspired by the same, we can have a superficial injection of drugs and vaccines through these micro needles without much of pain. So they don't actually they don't inject deep into the skin where the pain receptors are present. So they will be very superficial. So they don't stimulate the pain receptors. And once they are injected, they slowly dissolve and release the molecules into the skin. And coming to back to microfluidics, we people have adapted the channels that are present in the nostrils of a dog. A dog actually can sense very very small quantities of gaseous materials present in the atmosphere. Right, it can sniff better than a human. That is because it has the channels within its nostrils, where it has so many channels that there is a lot of surface area that is present for the gaseous molecules to bind and get sensed. The same way in microfluidics, you can have channels, micro channels, and you can have different molecules bind to those channels. And in those fluidic, microfluidic channels, you can have detection, detecting sensors that are nanoparticles here, which are silver nanoparticles, gold nanoparticles, and other nanoparticles, which can actually give out something called Raman signal, where where we can have an enhanced Raman signal from when we have silver nanoparticles bound to a gaseous molecule. So very specifically at a particular wavelength, we can excite these sensors, and we can get surface signal, surface enhanced Raman signal, and we can actually have very very small sensing ability, that is a single molecule sensing ability with these surface signal molecules. Apart from this, we nanoparticles could be used in various applications: information technology, energy, as I said, medicines, and consumer goods. So consumer goods, when we look at L'Oreal, all these cosmetics have titanium oxide or zinc oxide in their products. The basic idea is they actually can absorb a lot of UV very efficiently. So when you put them on your face or in your hand, when you venture out in sun, you can avoid UV to penetrate into the skin. These cosmetics can actually absorb all the UV light present in the sun and will not affect your skin. So this comes, brings to the end of our nature and nanotechnology part of the talk, and that particular section I wanted to give so that I don't want to straight away go into some science and research. It will be a bit boring, and I don't know even the first part by itself how much stimulating it was to the general audience, the undergraduates and postgraduates. But the basic idea of that talk was to get inspired from the surrounding that you have, from the leaves, the insects, from your own body, how nanotechnology is present everywhere around you. It is not just about some synthetic lab-made material that only when you go into a lab you will be able to see. No, You just can't see them, but it is there everywhere. And if you get inspired by such mechanisms, you will have much more interest in taking up nanotechnology as a career, as a science career, or you can look at that as a inspiring career that can actually bring out a lot of change in biomedical and clinical applications of these technologies. Now, moving on to a little bit, not too much of the work that we do at our lab. I'm going to give just couple of examples of work that we have done. So we do a lot of drug delivery with nanoparticles, and to give why we need nano drug delivery. To give a very simple idea about what is exactly nano drug delivery. So in this slide, you see a healthy person with a headache taking aspirin, and the other side you have a cancer patient which who takes a chemotherapeutic drug called doxorubicin. So when we are looking at this healthy patient, he has headache, which is very normal to any human being, right? And when we have a very severe headache, which doesn't go even with sleep or regular diet, you or with rest, you end up taking a molecule, a drug, which is normal aspirin we take. And of course, we take it orally, not injection. I just gave that as a, um, you know, an illustration, but then you end up taking it orally and it gets absorbed in your intestine. It goes into your systemic circulation, in your bloodstream. And when it goes into your bloodstream, it goes everywhere, right? It goes everywhere. And if you inject it also, it goes everywhere. It directly goes into the bloodstream. And when a part of this area also receives some amount of this aspirin. So let's say you are taking 500 mg of aspirin. Only a fraction of it reaches the brain. Everything else moves across the body because it is a very small molecule. It can go to nook and corner of our body. So this fraction of aspirin that reaches the head is good enough to have some relief from your headache, right? So you take 500 mg and you have, let's say, 50 or 100 mg reach there, and the headache is gone. And rest of the 400 mg, let's assume, is going everywhere in our body. But aspirin being a very specific molecule which acts only on pain receptors or pain causing mechanisms, which is COX-2 signaling pathway. So it doesn't affect any other mechanisms or functioning of our body. Now, after some time, this aspirin, once its work is done, it gets excreted in the from the body, right? But in contrast, when we take a cancer patient, the same thing happens. In case of cancer patient, most of the time these molecules are not orally taken. They are actually injected. Very few molecules are orally taken, and when they are injected, let's just take injection as one of the way of injection as the way of administration. We end up injecting it into the cancer patient, and the tumor, because it is very fast growing, it takes up a lot of doxorubicin, and it tries to consume it because it is very selfish, that it wants to consume a lot, not giving enough nutrients to the rest of the body. So it grows faster because it consumes a lot. So along with the regular nutrients, it takes up a lot of this doxorubicin also that is present in the blood. So it accumulates a lot of doxorubicin here. But that doesn't mean 100% of the injected drug goes to the tumor. A major fraction of it goes to the rest of the body as well. In contrast to aspirin, doxorubicin is not safe. it actually binds to the DNA of every single cell, and it breaks, it damages the DNA, right? So when DNA is damaged, obviously I'm talking. So obviously it's not going to be easy on the cell. It will be it is going to be very detrimental. The cell is going to go into apoptosis. So killing the cell with doxorubicin is good if the cell is present inside the tumor. But if the cell is elsewhere in the body, and if that cell is going to be affected by doxorubicin, it is not good for our body, right? So that is where the nanoparticles comes in. Can we actually avoid the systemic distribution of the drug? And can we actually deliver the drug only to the tumor here? So moving to side effects of, yeah. So when we moving to the side effects of the chemotherapy, when a cancer patient takes a drug, something like some drug like doxorubicin, you will see the cancer patient lose a lot of hair. They will lose a lot of hair. They will lose a lot of muscle. And others really high accumulates these drugs, and they are going to be these sides of our body are going to be affected much more than rest of our body. So you will see a lot of hair loss. You will see a lot of pain, new mouth sores, that is open wounds that are present in our that then happen in our mouth. A person will have breathing because the lung will not be able to function properly. So the immune system in general will be affected. So a lot of issues that will come with regular conventional chemotherapy. But in case of nano drug, because the rest of the body delivers only to the tumor, it will avoid any of the side effects. It can actually improve drug solubility. As I said, oral delivery of these drugs is not possible because most of these drugs are hydrophobic. They will not be soluble in water. So they will have to be solubilized with some stabilizers, and they will have to be directly injected into the blood. So that is also not good. You end up adding more and more toxic components just to improve the drug's solubility, and you inject it into the blood. And nanoparticles will improve the drug solubility. It will keep the drug safe inside the matrix of the nanoparticle, and it will improve the bioavailability and pharmacokinetics. That is, it will improve the availability of the drug at the tumor site, and it will also improve the drug's uh, residence or presence inside the body much more for much more time. And it will improve the therapeutic outcome. Of course, it will shrink the tumor, and it will avoid the side effects, and it can also control the amount of drug that is released over time. So based on these concepts, we have developed some of the nano formulations of for drug delivery. One of the example is we have made this polymeric nanoparticles, which can actually encapsulate two drugs. One is doxorubicin, and another one is docetaxel. And these two drugs are compartmentalized in the core and the shell, basically. So you can see in this electron micrograph, there is a core and there is a shell. And the core contains docetaxel and the shell contains doxorubicin. Doxorubicin being a fluorescent molecule. When we made microparticles of these core shell particles, we were able to see in the shell the doxorubicin was light yellow, right? And because we have the shell is made up of something called carboxymethyl chitosan, which has both carboxyl group and the amine group, and it can actually provide something like it can provide pH mediated switching of the surface charge. So you will see that at acidic pH it is positively charged, and in the alkaline pH it is negatively charged. Alkaline, neutral to alkaline actually, it is negatively charged, and it has an, so it obviously will be having an isoelectric pH around 5 to 6. So this inside the cell we have something called a lysosome, which has a pH of about 4. Outside the cell, the tumor pH will be about 6 to 7. So it has a negatively charged surface charge before getting into the cell. Once it gets into the cell, it switches into positive charge, and this positive charge actually helps in something called it helps in aggregation and also called lysosomal escape into the cytoplasm, and it releases the drug into the cell. So these particles were able to provide significant toxicity compared to in different cancer cells, breast cancer, prostate cancer, and lung cancer cells. And the combination was able to provide a better outcome compared to single drug particles and single free drugs. So you see a combination of two drugs will have a better outcome than single drug regime. It is very important because in the clinical setting as well, we have multiple drugs that are given to a patient so that there is a better clinical outcome. So this dual drug combinatorial nanomedicines are something that we want to realize a bit closer. Apart from this, apart from regular chemotherapy for cancer, we can also encapsulate different molecules which are present from which are extracts of natural leaves and herbs, which otherwise do not have a good soluble water solubility, or they may not have a good bioavailability. That is, when they are taken normally, these extracts of leaves and herbs are taken orally, right? And most of the time they don't get absorbed in our intestine, and they don't actually get into the bloodstream to the concentration that is actually required for a therapeutic outcome. So here, taking COVID as an example, last year we tried to, this is basically a computational study, but we wanted to understand how many of the molecules which are showing very good promise against SARS coronavirus, but what are their pharmacological properties, that what is their bioavailability, what is their solubility. So we ended up finding that there are many molecules which can actually effectively bind with proteins that are present in SARS-CoV in the virus. But the issue is they are all taken orally and they don't get absorbed. They actually get degraded in the intestine, and they end up not delivered into the bloodstream as much as they are required to be. So you see, I'm sorry, you see, this molecule delphinidin is something that is present in pomegranate, which is in Madulai, right? So it is one of the molecule that gives the red color in pomegranate, and it is one of the top hits that we found computationally, that they were able to bind to the important main protease enzyme required for viral assembly, viral particle assembly, inside the host cell for the coronavirus to multiply inside the host cell. So it was able to effectively inhibit computationally. We were able to find, but it if you consume a lot of these, it is not going to be that much effective against SARS-CoV, because it will get degraded in the body. And for such molecules, you can directly encapsulate them into nanoparticles, and you can have different types of application into the body through nasal inhalation or through oral injection as well, or through systemic administration. You can deliver these very potential drugs that are naturally available. You don't have to synthetically make them, and have a better outcome, especially in conditions such as COVID. So moving on from chemotherapy, drug delivery, in our group also works on something called photothermal therapy. So this is related to photonics, where I was talking about gold nanoparticles, how they different shapes have different absorptions, and how they can be used for sensing applications, Raman signals, how it can enhance Raman signals. And apart from that, in case of therapeutics, they can be used for something called photothermal therapy. So prior to going into the technology, we need to know who invented it. And I show this slide in most of my talks, where you have two female researchers who brought together their expertise to invent a new technology for cancer therapy. And Naomi Hallis in Rice University, she invented something called gold nanoshells. So she invented that for some photonics and photo engineering applications. But then when she put that together, the idea together with Jennifer West at Rice University again, they could actually converge the technology for biological applications. So I should be thanking another female who taught me about photothermal therapy and nanoshells, Professor Deepthi Menon at Amrita Center for Nanosciences. And the basic principle behind this photothermal therapy is, when gold nanoparticles, when they actually absorb light energy is an electromagnetic radiation which has electrical and magnetic components, right? So this electric component, when it interacts with the gold being a metal, it has free electrons moving around very randomly. As you know, it is so conductive, right? So this electron cloud ends up getting polarized. It comes to the surface, and this cloud keeps on switching when when the incoming electric field switches, right? It is a alternating field. When the field switches, the polarization, the dipole that is formed, the electron at one side and the other side, which doesn't have or devoid of electron, is something similar to holes in the semiconductors. So a dipole which is formed, it keeps flipping with the change in the electric field, the field direction. So when the flipping actually happens at a very high frequency, it can absorb the frequency. When the frequency of the flipping of the dipole matches with the frequency of the incoming light, when it matches, there is a lot of energy that is absorbed by these nanoparticles from the light. Okay, so this energy that is absorbed later is given out as thermal energy. So that is where the photothermal therapy comes in. So photons comes in contact with the gold nanoparticles, and when the energy that is absorbed by the gold nanoparticles is released as thermal energy. So photothermal technology which comes out of it. And Hallis in her pioneering paper published in 1998, she proposed or she devised these nanoshells where she took silica core and she coated gold shell on top of the silica core, and the thickness of the shell can actually give rise to different absorbance. As you can see here, the thickness, if it is 20 nanometer, it is more, the lambda max is more towards the 700, but when it becomes, the core to shell ratio is increasing, that is, the shell becomes smaller and smaller, the absorbance becomes is shifted towards the NIR region. It gets shifted, and you can have absorbance in both near infrared 1 and 2. So combining this technology with for biological applications, along with Jennifer West, Naomi has published this paper in PNAS, Proceedings in National Academy of Sciences. So I think in 1999 or 2000. So they could see that when these nanoshells are incubated with the cancer cells, and when the nanoshells are incubated with the cancer cells and the laser light is incidented, you'll see that only the laser incidented area has killed the cells, but rest of the area has not. So only when the nanoshells and laser combine together, they produce this photothermal effect. And laser alone, of course, is harmless, and nanoshell is also harmless. As you can see, the surrounding area is not affected. So nanoshell, without the laser's interaction with the laser, it doesn't actually kill the cancer. So this basically is the panel which shows dead cells. So the fluorescein, dextran, can stain only dead cells. So there is no dead cell in the laser only study, and whatever the dead cell that were present here at the center is shown up here. So we ended up seeing live cells here and dead cells here. So only when these two combine together, you see cell death. So as I said earlier, because it is absorbing in NIR region, the laser is also NIR laser, and NIR laser has much better depth, penetration into the skin. And based on this technology, Naomi Hallis had a lot of patents for photothermal agents, medical imaging agents, and Raman sensing agents. So they were able to start a phase 2 clinical trial on these nanoshells for photothermal therapy of head and neck cancer, prostate cancer, and lung. So nanoshells, they made with silica as a core, but it can be made with polymeric core as well. And you can use polymers, and you can actually functionalize the nanoshell through two various ways, various pathways. So here you are seeing we had made a gold nanoshell by seed mediated method. In case of here, it is chitosan coated, so seeds absorb onto chitosan and eventually gets functionalized as a nanoshell. Here there is it is not our work. It is a work there where they have used a ligand which has thiol group. Thiol is attracting towards gold nanoparticles, so you end up attaching gold and forming gold nanoshells. And apart from full shell, you can also make something called half shells if you have these these polymeric nanoparticles on a substrate, and you sputter gold on top of it. So only the top area of the gold polymer, polymer will get deposited with gold. So when you, after the sputtering, you remove the particle, you will see the top layer having gold, and the bottom layer, because it is hidden from the sputtering, it wouldn't have any gold attached to it. So you will end up having half shells. So even this half shells are photothermally active. So this is just to give you an idea there are various ways you can prepare polymeric particles and gold nanoshells similar to silica nanoparticles and gold nanoshells. And we have made these gold nanoshells on polymeric particles the same way, whatever I told you. And you can see these nanoshells under TEM. And coming directly to their testing in tumor studies, so here, the first author, Dr. Deepak Chauhan, he was able to make nanoshells which can absorb at 750 nanometer, and which can absorb and another nanoshell which broadly absorbs with a lambda max at 950 nanometer. So we found the 750 was much more effective in generating heat when the laser is incidental on it. And you can see the amount of temperature here is closer to 60 when when it is 750 nanometer absorbing nanoshells. And if we take nanoshells which are absorbing at 915, the amount of temperature that was generated is less than 45. And in the animal studies, it was found that compared to control is basically you see these are tumor cells which are injected under the skin of a mice. So these tumor cells are inherently fluorescent. So they have a protein which is fluorescent protein. So when they are alive, this fluorescent protein gets secreted, and there will be a lot of fluorescence coming out of it. So when these cells multiply, the fluorescence actually, the tumor becomes bigger, and the fluorescence intensity also becomes much more, right? So when the nanoshells are injected into these mice containing the tumor, you see only the laser on the tumor doesn't make much of a difference. It is something similar to the control, which is only saline or without any nanoparticle or anything, which is just saline or PBS. In case of both 750 nanometer laser and 915 nanometer laser, there is not much of a change in the tumor growth. In fact, the intensity of the fluorescence has increased. But when we are looking at nanoparticles along with the laser, the the tumor actually has shrunk, and because it has shrunk, the luminescence is also lost, the fluorescence is also lost. So in this particular group, 750 nanometer group, even after 6 months, the tumor did not come back. Okay, but when we are looking at 915 nanometer laser group, the tumor actually shrunk to a little bit. There was decrease in the fluorescence, but after the treatment, it grew back into a bigger tumor again. In fact, much bigger than the initial tumor. So the 750 nanometer laser was able to provide better photothermal effect on the tumor, and it was also able to completely kill all the tumor cells that it did not come back. So the final slide shows how we are looking at something called EPR to get these nanoshells accumulate in the tumor. What is EPR? EPR is enhanced permeability and retention. So as I said in my initial slide, tumor basically is a selfish mass of cells within our body, our own body. So it wants to eat a lot, and at the end what it does is it will have a lot of blood supply around it, because it needs to eat a lot. It wants a lot of blood vessels to supply nutrients to it. So it ends up taking up all the nutrients. So because there is a lot of blood supply to the tumor, the nanoparticles present in the blood accumulate more in tumor than elsewhere, like similar to the drug. But in case of drug, it is a very, very small molecule. it can actually percolate very easily into different blood vessels and go into different tissues. But nanoparticles are more than 100 nanometers or around 100 nanometers or 200 nanometers in size. It cannot diffuse into tissues as easily as a molecule. So you end up we are in this last slide, so we'll finish with this slide. We end up accumulating more in this tumor through something called EPR, enhanced permeation. That is, the blood vessels are having a lot of very small pores, and it gets a lot of these nanoparticles moving into the pores and getting into the tumor and accumulate in the tumor. So enhanced permeation, it permeates well into the tumor. And retention, as much as he eats, it doesn't actually gives out the waste. So regularly in normal tissue, the nutrients are taken up, but it also, the waste is also given back into the bloodstream so that it is excreted out. But in tumor, all these waste accumulate. So whatever is taken in is kept inside. It keeps accumulating inside. So the nanoshells, whatever we saw in the previous slide, accumulates more in the tumor through this EPR effect. In fact, it accumulates through this effect more. Even with receptor targeting, it accumulates lesser. So EPR is much more effective way of accumulating in tumor than receptor targeting mediated accumulation. So this is something that it was exciting results that we found in that particular work. So I might have been a bit lengthier in explaining things, but I hope I didn't make the audience fall asleep. So before I could finish, I need to acknowledge all these persons who were able to give who were able to provide good collaborations, and all these funding agencies which was able to give enough money to have some work done at our laboratories at INST. So thank you. So thanks for listening. I'm open to questions. Hello. Hello.
Thank you, sir. Am I audible to you?
Hello.
Sir, am I audible to you?
ഓക്കെ ഇപ്പോൾ തീർത്തു കഴിഞ്ഞു. ഓക്കെ ഓക്കെ ഓൾ റൈറ്റ്. ഞാനിപ്പോൾ തിരിച്ച് പിന്നെയും ജോയിൻ ചെയ്യാം. ഓക്കെ.
There's a small technical issue. We'll be returning back very soon. Participants, please bear with us. ഞാൻ ലീവായോ? ലൈക്ക് ഞാൻ We just very sorry. There is a small technical error in the connection, I guess, and I think it was a very wonderful session, and I really thank Dr. Asif Khan Shanavas because that was definitely refreshing to actually see our textbook concepts like van der Waals forces and things like that being put to use in a field like medicine. So. Now we'll wind up the session. So I invite Dr. Joe Kuyakuden, Assistant Professor, Department of Physics, St. Thomas College, Thrissur, for the vote of thanks.
Thank you, Anupama. Thank you, Dr. Asif Khan, sir, for the nice and informative session. The topic was natural and synthetic nanostructures application in healthcare. He started the webinar by explaining the fundamentals of nanoscience in simple language, then to the Nobel Prize winners of nanomolecular machines, its structures, and its working principle, its application to curing our health issues, and then the methods used to make nanomolecular machines. He also discussed about manifestation of the nanoscale size effects, including its physical, chemical, optical properties, etc. He also mentioned about the nanostructures in creatures found in nature like nanotoes in beetle, fly, spider, gecko, etc. He also explained about the applications of nanotechnology in various fields. Thank you, sir. On the behalf of Department of Physics, St. Thomas College, Thrissur, I express my sincere gratitude to Dr. Asif Khan as he accepted our invitation to the webinar, even in his busy schedule, and also to share the valuable information to us. Thank you, sir. I also express sincere thanks to our beloved principal, Dr. K.L. Joy sir, for his valuable presence in this session. I express heartfelt thanks to our HOD, PM Joy sir, and all other faculty members, especially Dr. John Snudworth, as he is the coordinator of the program, and to our dear students who attended the session. So once again, thank you to all. Thank you.
Thank you, sir. We deeply regret that we couldn't do a Q&A session after the lecture, but thank you once again, thank you, Dr. Asif Khan Shanavas, sir, for that wonderful lecture. And we also thank all the participants who have bared with us and participated through the entire length of the program. The feedback link is posted in the chat box. Please kindly do fill it.